Hello boys, welcome back to another session of online biology video tutorials, and this session is meant for class 12th biology students, and this is for 28th July 2021. We are entering into an all important, very curious, interesting chapter number 5, Principles of Inheritance and Variation. This chapter is very interesting because of two reasons. Reason number one, it gives an answer for us why we are what we are. I repeat, why we are what we are. And second, the curious question that always lingers in our mind is, what would be our future generations? How do they look like? What are the characteristics that they would be displaying? Are the characteristics that are present now in present generation, will they be transforming to future generations as such, or with slight modifications do they go? So n number of questions that arise, that linger in our mind, would be answered in this chapter. And not to forget, sufferings due to genetic diseases. Unfortunately, some of us suffer because of no fault of ours, but because of our birth. Why should it be? So we are going to answer such questions in this chapter. That's the reason why I said this chapter is not only important for the sake of CBSE board exam, NEET exam, UPSC ND exam. It answers all that we experience in our life. Hence, very interesting chapter. With that note, we go ahead. Principles of inheritance and variation. Thanks to this gentleman, Gregor John Mendel. He is referred as father of genetics. And whatever we are going to study, whatever research that is taking place worldwide today about the genes, it's all because of the inspiration given by this gentleman. That's why we say he is father of genetics, Gregor John Mendel. And that's why, to honor his name, we are calling it as Mendel's laws of inheritance. Okay, and let's see what are those. And before we go actually into the laws, Mendelian laws, let's understand some of the technical words that may arise in this chapter. First, genetics. It is the branch of biology that deals with the study of heredity and variation of characters, or we say study of genes and chromosomes. So genetics is branch of biology that allows us to study genes and chromosomes. Next, inheritance. Inheritance, transmission of characters from parents to offsprings, and this transmission is based on heredity. So inheritance is basically transmission of characters. And what about variation? Variation is the difference between parents and offsprings. So you can see the figure of Malayalam actor Mammootty and his son. So variations, differences are there between the senior Mammootty and junior Mammootty. And characters, a heritable feature among the parents and the offspring. For example, eye color. Eye color is a character. And trait, variants of a character. Sometimes people confuse between character and trait. Let me make it clear. Character is a heritable feature, like eye color. What is that color? That speaks of trait. Trait speaks of the color, like brown eye, blue eye, black eye. So variations that are present in a character are traits. Like you are seeing, some of us will be having brown colored eye, some of us will be having blue colored, some of us gray colored, even green colored. And then alleles. Again, this word comes often. But before we go for the alleles, we must know what is a gene. Gene is the inherited factor. Inherited means transmission from parents to offsprings. Something from our parents has come to us. That's a gene. So gene is the inherited factor that determines biological characters of an organism. Whatever we exhibit both inside and outside, thanks to the genes. And from where we are getting these genes? From our parents. So if we study those genes that are present in us, that are responsible for our characters and traits, is genetics. So study of genes is genetics. And heredity is the study of transmission of characters from parents to offsprings or from one generation to the next. The characters that are passed from one generation to the other are called hereditary characters. And variations may be defined as differences in characteristics shown by individuals or species and also by the offsprings or siblings of the same parents. So, from that point of view, the word comes allele, that is alternative forms of a gene. Alleles are alternative forms of gene. For example, capital T, small t. Capital T gene is responsible for tallness of the organism, like tall human being. Small t is responsible for dwarfism, shortness, stunted growth. So these two are, capital T and small t, are alleles of a gene for the character height. So basically one gene is there, that gene is responsible for height of a person. And the variants are, the height may be short, the height may be tall. So these variants of the gene are alleles, right? Now we go to next one, homozygous condition. It's a condition in which chromosome pair carries similar alleles of a gene. See, similar alleles. We said capital T, small t, like that. So these are similar. Also known as pure lines or true breeding. For example, capital T means pure tall. There is no doubt in that organism becoming tall. Small t, again pure dwarfism. This person cannot become tall. Capital Y, and small y. Here even in the diagram you can see homozygous condition, presence of B. Okay, capital B. The variation of that is heterozygous. Hetero means different. Different alleles are there. And the difference in the alleles is because of dissimilarity. So the condition in which a chromosome pair carries dissimilar alleles. Look at that, capital B, small b. Dissimilar alleles. Then we say heterozygous condition. So example, capital T, capital Y, small y. So basically homozygous condition, organisms have two similar genes or alleles for a particular character in a homozygous pair of chromosomes. That means it can be capital T or small t. Here, do not think that there will be a gene in the shape of capital T or in the name of capital T. For convenience sake, we are taking that as example, right? And since we are talking of height, the variations of height is tallness. So we are picking up that first letter of the spelling, tallness. That's the reason why we are saying capital T or small t. And do not assume that there will be a gene called capital T, or do not assume that there will be a gene with the shape of capital T, alphabet capital T, right? Now, it is the organism that containing two different alleles or an individual containing both dominant and recessive genes of an allelic pair. Say dominant is capital T, small t is recessive. So such a condition, if genes are there, the chromosomes are carrying, then we say heterozygous, dissimilar alleles, capital T and small t, right? You can see same thing, homozygous condition, heterozygous condition and homozygous Capital B and small b are two alleles of a character. Further, dominant character. You have seen sometimes people are, you have observed in your own family, in your own native place, whenever people group together, one or two persons in the group will dominate. They will not allow others to talk, only they will be talking. Or their appearance will be very dominant, very tall and hefty in the group. So, this dominance character is because of the gene again. The character which is expressed in heterozygous condition, it indicates with capital letter. For example, capital T, small t, just now we said. So here, capital T is called dominant gene and small t is called recessive gene. Though recessive gene is present, capital T is able to express itself. So the plant becomes very tall or person becomes very tall. So capital T henceforth is referred as dominant gene and the character that it expresses like tallness is dominant character. Like we have got here cleft chin, right? And not having that smooth one is recessive. Similarly, widow speak, okay, this and no widow speak, recessive character. Dimples in the cheek, dominant character. No dimples, recessive character. Brown or black hair, dominant character. Blonde hair is recessive character. And then freckles, dominant character. No freckles is recessive character. Brown eye is a dominant character. And gray or blue eye is recessive character. Free earlobe. Check your earlobe, whether your earlobe is attached or free. If you got a free earlobe, it's a dominant character. And in some cases, in some human beings, the earlobe is attached. If it is attached, it's a recessive character. So, recessive character is a character which is suppressed, means unable to express itself in the presence of a dominant. Of course, if both the alleles are recessive, like small t, then both express together. Okay. Only in the presence of dominant gene, recessive gene cannot express itself. So, recessive character cannot be expressed in the presence of a dominant gene. So, this can be only in heterozygous condition. It indicates with small letter. We said small t, for example. Phenotype. Phenotype is the physical expression of a character, like tallness, somebody being tall, somebody being hefty, someone with brown eyes. So these are all physical expressions. And genotype, on the other hand, is the genetic constitution of a character, or we say gene makeup or gene ratio, genetic composition, we can say. So genetic composition makes genotype, and that is responsible for the phenotype, right? Whatever genes that are present is genotype, and whatever genes that are present will make the individual to express corresponding characters. That's phenotype. And we often hear the word hybrid, an individual produced by mating of genetically unlike parents. Okay, so different. Like we always say a European marrying a West Indies girl, or vice versa, a West Indies person marrying a European girl. So we say these are hybrids, and similarly species. So again, thanks to this great man, we got John Mendel, father of genetics. He has done a lot of work, and as we go through this chapter, I am sure each one of you will appreciate the contributions of this great man, and we are thankful to him for whatever senses that he has given to us. And he has proposed some concepts, certain important points, which are referred to as Mendel's laws of inheritance. And what I personally feel is his patience. He has enormous patience like earth, the earthly patience that he had, and he conducted thousands and thousands of experiments. And mind you, he was a father in a church. He was father in a church, and being a father, remember, he was not a scientist. Only he was father in a church, In spite of being a father in church, he had interest in nature, he had interest in gardening, he had interest in plants, and that made him to discover all this. So Mendel conducted some hybridization experiments on garden pea. Now this usually UPSC exam. The scientific name of garden pea, Pisum sativum. And remember why I praised him? 7 years he conducted the experiments between 1856 to 1863. Remember, if a person has to do experiments for 7 long years, how much patience he should have had. And then, what are the steps in making a cross in pea? So what he has done is he has selected garden pea, and there are reasons for selecting garden pea, because he has conducted this basically because of quick completion of life cycle, and the number of chromosomes also is 14. Diploid condition is 14. So Mendel could identify contrasting characters in the garden pea. So the number of contrasting characters that are present in garden pea attracted Mendel to perform experiments on that. Like he could observe in his own garden around the church. The height is one trait he studied, and the varieties are tallness and dwarfness. So tall pea plants and short pea plants. So that's the character. Second character is flower position. He has observed that flowers in garden pea, some were axillary, means at the tip of the plant, and some are terminal on the sides. Then pod color, he could observe, even today we are observing green color or yellow color. Pod shape, it is fully inflated, round or wrinkled. And flower color, as you are seeing, violet color and white color. Look at that, variations in the flower character. And then seed shape, as you are seeing, some of them are round, some of them are wrinkled. And color, seed color also, most of the time you are seeing fresh one green and the old one is yellow. So varieties are there. This made Mendel to perform experiments on garden pea. So selection of two pea plants with contrasting characters, that is the first step he has taken. Second, he has done emasculation, removal of anthers of one plant to avoid self-pollination, as you are seeing here, emasculation, removal of the anthers with the help of a scissor, cut off that. And this is a female parent, of course. And then pollination, collection of pollen grains from the male parent and transferring to female parent. So it is intentional one. And collection and germination of seeds to produce offsprings, and see the varieties among the offsprings with reference to one character, two character, like that. And as I said just now, Mendel has selected garden pea because of 7 characters, because of the pea plant having contrasting characters like stem height, tall and dwarf, flower position, axial and terminal. You can see here tall plant, dwarf plant, axial flower and terminal flower. You can see flower color violet and white. Pod shape, it is inflated and constricted. Pod color green and yellow. Seed shape, round seeds, wrinkled seeds. And seed color, yellow color and green color. So because of the presence of contrasting characters, Mendel selected this. And inheritance of one gene, he conducted an experiment, monohybrid cross, wherein he has selected, why we are naming it as monohybrid, mono means one, and since it is the crossing, the resultant product is a hybrid, we are saying monohybrid cross between tall plant and dwarf plant. And when we say tall plant and dwarf plant, we are taking one character, that is height. That's why monohybrid cross. Mendel crossed tall and dwarf pea plants to study the inheritance of one gene. That's why mono, one character. So cross between yellow seeded plant and green seeded plant. He is taking only one character. So what he has done, he has taken first a plant, the parental generation, we call it P generation, homozygous tall, capital T, because it is homozygous, similar alleles. And since it is tall, the plant is tall, it is pure tall, capital T. And the homozygous dwarf, small t. When the cross has taken place, see the gamete that will be coming from this plant is capital T, from this plant is small t. So the combination of two gametes fuse together to form a zygote, and the zygote is becoming embryo is going to become a plant. So the fusion of gametes, capital T and small t, results in the plant having this genotype, and because of the presence of this genotype, the plant is becoming definitely tall like the father plant, but the genetic composition varies. In the parental one it is pure tall, capital T, whereas in offspring it is capital T, small t. So we say heterozygous tall. And when selfing takes place between the heterozygous plants, that is capital T, small t, the gametes that will be formed obviously is from this plant capital T and small t, from another parental plant also, since it is genotype is capital T, small t, it will be capital T and small t. So the fusion of these gametes like capital T and capital T is resulting in capital T pure tall, and the same zygote combines with this capital T, small t, still it is tall. Why? Because of presence of dominant gene capital T. And then small t, capital T, again the combination is capital T, small t. Again this plant also becomes tall because of presence of dominant gene. What is the difference between plant 1 and plant 2, 3? Plant 1 is homozygous tall, plant 2 and 3 is heterozygously tall. And of course last combination is this small t, which will be pure dwarf. So when we put this genotype in the form of squares, this is discovered by a British scientist called Puneet. So we say this checkerboard way of representing the genotype as well as phenotype is called Punnett square method. And when we see the ratio in the monohybrid, phenotypic means physical appearance. How many tall plants are there? 1, 2, 3. So 3 tall. And how many short? Only 1. So 3:1 is the universal ratio of monohybrid cross. Though he has performed in pea plant, when it is applied to all, it can to know that any organism will be abiding by this. Exception will be there, of course, we will talk about the deviations, but predominantly it is phenotypic ratio is 3:1. And what about genotype? Homozygous tall, capital T, how many are there? Only 1. Heterozygous tall, capital T, small t, 2 are there, plant number 2 and plant number 3. And homozygous dwarf, only 1. So the genotypic ratio is 1:2:1. So in monohybrid cross, always the phenotypic ratio is 3:1, genotypic ratio is 1:2:1. So this is possible because of transfer of one gene. That's why we say inheritance of one gene. So Mendel made similar observations for other pairs of traits also. He proposed that some factors, of course, Mendel used the word factors because remember in 17th century, where was the technology improvement? There was not even a good hand lens. So he guessed that there must be something called factor which must be responsible for this. And these factors were inherited from parents to offsprings. And those factors which Mendel called, now we are calling it as genes. So in this you can see, I said a lot of patience for him because he could calculate even the mathematical numbers. How many dominant, how many recessive, when the combination is this, flower one gene inheritance. 705 plants, dominant